Start.
Dear students, on behalf of my college and the department, I welcome you for the 6th semester, and I am going to deal with paper 2. Its title is Genetics, Biotechnology and Nanotechnology, and we are supposed to complete this whole paper syllabus in 45 hours for the marks, that is 80. However, the beginning topics are like this, which I am going to engage. There are totally about 9 topics I am going to do. The first one is an introduction to genetics. Second is interaction of genes. Third is sex-linked inheritance. Fourth is sex determination. Then fifth is linkage and crossing over. Sixth is mutations. Seventh is human genetics and eugenics. And the one more, that is cytoplasmic inheritance, with the help of an example, that is paramecium or alien. However, today let us start with the first topic or the first chapter, that is introduction to genetics. I think you people are very much familiar with the terminology, that is genetics. Genetics is derived from gen in terminology, which means to become. I hope, I believe that you people have been referring this genetics subject since your SSLC standards, PUC standards, as well as here in degree, that is for the final year. However, don't be afraid of the quantum of the syllabus which is there. Let us go by the topics one by one by means of simplifying it. Maximum part of it is already studied by you people, and let me start with the first one. As I said, that genetics is a term derived from a Greek terminology, that is gen means to become, that is to after birth, how an individual species should develop, like of its own parent type or not. That is the main meaning or the actual meaning of this. I want to ask you one question, like why you people look like your father and mother? Why the newly born young individuals look like their own parents, both male and female? That is all because of one main phenomenon. That's what we call it as heredity. Here, heredity is a phenomenon of transmission of characters from parents to offspring. Offspring means newer generation, new young individuals. Here this heredity, I said that it is a phenomenon, and one more term I like to use, that is inheritance. Inheritance is a process, it is a process, whereas heredity is a phenomenon. Inheritance is a process of transmission but not transfer. Please remember the term that we need to use is it is a process of the transmission of characters from parents to their children, from parents to their offspring, from parents to their progeny. One more terminology, synonym, that is progeny. Okay? And now, what all is involved in learning genetics? This genetics is all about knowing two things. The first, as I said before, that it is a heredity, and one more is variations. What are variations? Now you know heredity, as the characters present in the parents are transferred through gametes to the next generation, to their kids or the offspring or the progeny. This is the main reason why we look like our both father and mother, and the same in all other sexually reproducing organism species. And genetics is to know about not only heredity, it is also to know about the variations. Look at it, the same thing that we are not the exact Xerox copies of our parents. In some ways we resemble our father, in some other ways we resemble our mother, and in other ways we do not resemble. They are something different. We look something different from our parents. This is because of the reason called as variations. Variations cause differences in between parents and offspring, which clearly indicates that in sexually reproducing organisms or individuals, definitely the newly born progeny or the offsprings resemble their parents, and at the same time they also have or possess and exhibit different characters. That's why we need to know in detail about the mechanism of heredity or the mechanism of inheritance and the occurrence of variations. Already you know the term variations in case of learning evolution. What is it? Variations are considered to be the raw materials for the occurrence of evolution. Here, through the process of speciation, usually what happens, the newer and newer individuals come up on this earth. However, let us keep that process of evolution apart and confine to the genetics. Also, I want to give you another example. Why? In case of, usually we refer one royal family, that is Raj Kapoor's family. You might have heard, you might be knowing, like Kareena Kapoor, Karishma Kapoor, their parent, Ranadhir Kapoor, their parent, that is Raj Kapoor, in turn his parent, father, that is Prithviraj Kapoor. Look at it, from Prithviraj Kapoor to this Karishma Kapoor and this Kareena Kapoor. Look at the similarities in case with respect to their color, skin color, eye color, then other facial features. Similarly, I want to quote one more example, that is Indira Gandhi family. That is in case of, look at it, presently we are having a person, the political leader, that is Rahul Gandhi. His father was Rajiv Gandhi. In turn, his father Firoz Gandhi. But look at the Firoz Gandhi and his wife called Indira Gandhi. Indira Gandhi facial features we can see it very clearly in case of present Priyanka Gandhi. Why this resemblance? This is all because this can be understood. This can be very clearly assessed by considering a fact like heredity and the variations. It is very much enthusiastic and interesting science that we need to know, and we are going to learn under different headings because there are a lot of the phenomena, there are a lot of the events that take place in case of only the sexually reproducing organisms. And after knowing this, we need to know how exactly the heredity occurs, how exactly the inheritance occurs. For that reason, we need to consider the heredity and variations. These both together are essential for the formation of species, any kind of species. These two are very much important. One is heredity, another one is variations. Then where does inheritance occurs? For this reason, we need to dig into the detailed account of it. The first one, usually the nucleus, you know, the nucleus is the center of cell, where it is also the center of nucleus. Why the nucleus? Because nucleus contains DNA as well as RNA. DNA, what is the DNA? You all know, but let us not study all that in detail, its structure, composition and all. But what we need to know is its functional significance. DNA stores the genetic information. How? Because where do you find DNA? Definitely that is found inside the nucleus. DNA is known to store the genetic information, means the genetic information of the parents, of both the male parent as well as the female parent. And exceptionally in case of viruses, RNA acts as a genetic material. RNA acts as the storage of parental characters. That is only in case of viruses. However, let us try to know how exactly DNA is able to store the genetic information. Because you know DNA is a double-stranded and formed of nucleotide sequences, and any segment of DNA can act as a gene. Here we come across the term called gene. What is a gene? You know that gene is a hereditary unit. That means it is a main cause for carrying the characters or information about the character from parents to the offspring. In that way, gene is found present as part, as a segment of DNA. In a DNA, there is a sequential arrangement of nucleotides. Any set of nucleotides segment in a DNA could act as a gene, and gene is the one which is capable of undergoing replication, then mutation and expression. These are the main three functions of gene. You take any gene in any species organism, it must be present as a part of DNA. The segment of that DNA must have a sequentially arranged nucleotides. Then that gene must be capable of undergoing replication, that means duplication. You know, during the cell division, maybe mitosis or meiosis, DNA has to undergo duplication or replication. I think in 4th standard you must have studied it. Please recall your memory. Second one is mutation. You know, I think you must be very much familiar with this terminology. Mutant variety, COVID-19. It is not COVID-19 today. It is the third or second mutant variety of the COVID-19. What do you mean by that? ಕನ್ನಡದಲ್ಲಿ ನಾವು ರೂಪಾಂತರ ಹೊಂದುವುದು ಅಂತ ಹೇಳ್ತೀವಿ. That means what happens, the RNA present in the virus undergoes sudden and unexpected change for its better adaptation, for its better survival. That's whatever it may be. However, gene is capable of undergoing the sudden unexpected change for the survival of the individual. At the same time, gene is also responsible for expression. What do you mean by that expression? How do we say that gene is expressed? How do we see that gene is expressed? Say and see, look at it. Though it is present as part of ultrastructure of DNA, this gene is expressed in the form of specific protein molecule. We may be having thousands of characters inside and outside. Behind of every character, there is a gene expressed. And that gene is expressed, a character, through a protein molecule. That means fully our body is having characters with respect to their specific protein molecule. And that respective protein molecule is specifically encoded and synthesized by a DNA through a specific process that is called as protein biosynthesis. We will come to that later on. So this is how exactly we need to understand the process of inheritance or the phenomenon of heredity lies in the DNA, which you find it inside the nucleus of a cell of an individual. So this is how we can understand a gene. Then how many genes we carry in our cell? It depends, and we will discuss about it in the later part of this topic. After knowing this, what we need to know, what we need to study now, that is regarding the chromosomes. So here, chromosomes. ಕನ್ನಡದೊಳಗೆ ನಾವು ವರ್ಣತಂತುಗಳು ಅಂತ ಕರೀತೀವಿ. DNA, as we know, how much amount? Usually it is calibrated in terms of picograms, and this DNA appears, it exists inside the nucleus of a cell, we know, but it appears in the form of chromosomes. That means chromosomes are formed of DNA material along with some supporting proteins or the supporting protein molecules. However, these chromosomes are visible inside the nucleus of a living cell, where it is found very much coiled and condensed when cell is undergoing division, and it appears uncoiled and thin during the interphase. As you have studied the cell cycle, during the interphase, the nucleus of a cell shows the presence of very much thin strands, and that is nothing but the chromosomes. You must have also studied the ultrastructure of chromosome in your 4th semester. You just brush up your memory. You will come to know, or you will be able to visualize it. And here, these chromosomes are nothing but the DNA. DNA are nothing but the genes. And how many number of genes depends on the quantity of DNA material present inside the nucleus of a cell, living cell. And let us try to know the types of chromosomes: autosomes and allosomes. What do you mean by autosomes? Autosomes are the chromosomes that carry genes. Autosomes are the chromosomes that carry genes for the expression of characters. That is somatic characters. What do you mean by somatic characters? Somatic characters are the ones that are visible for naked eyes with respect to every living individual, and they work under our control. Look at your somatic characters are like your skin color, then they are present commonly in both male and the females. Color of the skin, color of the eye, color of the hair, then nature of the skin, then nature of the hair, then the number of digits with palm, number of digits with your foot, number of the limbs, paired limbs as hands and another paired limbs as legs. Likewise, and so on. All these characters are somatic characters, and they are commonly present in both male and the female individuals, by which we cannot differentiate that this is a male or this is female. They are all called as somatic characters. And such type of somatic characters are expressed by the genes that are present only on the kind of chromosomes. They are known as autosomes. I hope you followed it. Once again I repeat. Autosomes are the chromosomes that carry genes for the expression of somatic characters like color of your eyes, color of hair, color of skin, number of digits, then the shape of the nose, facial characters, whatever. These are all called somatic characters. And similarly, they are similar in both male and the female genders. Whereas allosomes are another set of chromosomes. They carry more number of genes for the expressions of sexual characters. Look at it. Sexual characters means even by looking at the shadow of the male or the female individual, we could come to know that this is a male individual and this is a female individual. By looking at the individual, how do we differentiate that this is a male sex and this is a female sex? By considering their sexual characters, reproductive characters. So they are all expressed by the genes that are present only on allosomes. So that always autosomes number is greater than the number of allosomes in every living species that is sexually reproducing type. The reproducing type. Okay, and this is about the chromosomes. There is one more term. We need to know few important terminologies that we come across in learning genetics. And let me give you the list of terminologies that we need to refer, that we need to understand. I write them one by one and I will explain. The first one, what we mean by trait, then terminology, that is a gene, already you know it. Third, that is allele. Fourth, chromosome, already you know that. Then autosomes, allosomes over. Then haploid. Then a diploid. Then phenotype. Then genotype. Still there are many more. We will understand first these terminologies. So, trait. What do you mean by trait? Trait is a character. It may be any character, either external or internal. That is, for example, let me say that every living individual, maybe an animal or a plant, must be having a character like height. You know, height could be expressed as a tall or a dwarf. So, like how we need to use the term as trait. Trait means here it is a height. Character means height is expressed either in the form of tall or in the form of dwarf. I hope now you follow the meaning of trait. Next one, gene. Already you know the meaning of gene as a hereditary unit, as structurally it is as a segment of nucleotide sequence in DNA and capable of undergoing replication, mutation and expression, and very much commonly known as a hereditary unit. Hereditary unit. Here we find this hereditary unit. It is present as a part of DNA. DNA is present as part of chromosome. You know, that's why chromosomes are often referred as vehicles of heredity. Vehicles are different, units are different. Units are genes. Genes are transported by the structures called as chromosomes. That's why they are referred as vehicles. Next one, allele. What is allele? One of either two or more alternative expressions. Alternative expressions. Look at it. One means you need to consider the chromosomes to be present inside the nucleus of a cell in the form of pair chromosomes, that we say that paired chromosomes. And here, to say chromosome, to understand allele, let me say when these two are the chromosomes. Here on this chromosome, what we find are the genes. This is conventional diagram, please don't mean it literally. And here, for expressing any one character, it needs at least two alleles. For example, here is a gene and here is also a gene for the expression of trait called height. Look at it. Here, gene for height must be present on the first chromosome, that is, we say that it is a paternal chromosome, and another one, let us say, represent it as maternal chromosome. Paternal refers to male parent chromosome, and maternal means that is a female parental chromosome. And here are the genes for expression of one trait called as height. Look at it, minimum 2 genes are required. So minimum 2 alleles are required. That's why I say that one of the two, any one of the two will express, not both the alleles will express. This is one allele for height, this is another allele for height present on paternal chromosome and maternal chromosome. ಅಂದ್ರೆ father chromosome ಮತ್ತು mother chromosome ಅಂತ ನೀವು ತಿಳ್ಕೊಳ್ಬಹುದು. ಈ ಎರಡು ಏನಂತೀವಲ್ಲ ಇವಾಗ ನಾವು alleles ಅಂತ ಕರೀತೀವಿ. Sometimes alleles are referred as a synonyms of a gene, but literally we need to understand categorically that each gene is expressing a character. That means behind of it any one of its allele must express. Usually for the expression of one character, allele, either 2 alleles or more than 2 alleles are essential. So that's how you need to understand. Allele or allelomorph is an alternative expression of a gene, so that for a particular trait or for a particular character. I hope you understood this. The next one, haploid and diploid. You people are very much familiar with this, where a single cell having a nucleus carrying the chromosomes, that is n number of chromosomes. N number of chromosomes. For example, it is a human cell. Human cell. It is carrying the chromosomes. If it is a 23, n means number of chromosomes. If it is carrying 23 chromosomes inside the nucleus, then it is said to be haploid. If the same human cell is carrying 46 chromosomes, then it is diploid. You understood? 46 means 2n, means n plus n is equal to 2n. N refers to single set of chromosomes. I said here are 46, means out of 46, 23 plus 23. Look at it. This is n and this is another n. So this is said to be diploid. If the nucleus of any living cell is carrying 2n number of chromosomes, then it is diploid, and n number of chromosomes, then it is haploid. For example, I said this is a human cell. If it is carrying only 23 chromosomes, means it is haploid, and if it is carrying 23 plus 23 means 2n number of chromosomes, then it is diploid. I hope you followed. And now I want to tell you how the haploid cells are formed. Haploid cells are formed through the kind of cell division called as meiosis. You know, meiosis is a kind of fractional cell division. For example, in case of a diploid human cell, that is most probably a cell of testis or cell of ovary in case of male or female respectively. And when it undergoes meiosis, it will form the daughter cells with the only n number of chromosomes. That's why it's called as a fractional cell division. And such type of cells are called as the haploid cells. In case of the animal species, haploid cells are usually either the sperm or the egg or the ovum. Similarly, in case of plants, you can refer like that is haploid cells could be pollens or the pollen grains and the ovules. This is in case of angiospermic plants, I am saying. But however, in case of animals, haploid cells could be either produced by male as sperms and ovum or ova produced by the female individual, female human being. So that's how we can understand what we mean by haploid cell as well as the diploid cell. And here for understanding genetics, what is more important is to go with haploid cells, sperms in male and eggs in female. Why? They contain n number of chromosomes, and if that is the case, how that n number of chromosomes get assorted, get distributed, we will see it in the next part of this topic. Next terminology, phenotype. What do you mean by phenotype? Phenotype means it is external morphological feature of an living individual. For example, if you think of an elephant, look at it. Elephant image will be formed in your mind. Look at it, how the body, very big, then having a face with a trunk, then a short tail back, then huge body, big size limbs. Look at it. All these are morphological features. And these morphological features are nothing but phenotypical characters. For example, if you consider me, am I tall or dwarf? I am neither tall nor dwarf, in between. Look at it. This is the way how exactly the height of an individual, color of a skin, color of an eye, then other facial features that are visible to you people, and my hands and the fingers, etc., they are all phenotypical characters. Then what do you mean by genotype? Look at it. Each phenotypical character is expressed because of its respective gene inside present on the chromosome as part of DNA, and that is known as genotype. Let me define phenotype. What do you mean by that? Phenotype is the externally appearing morphological character which could be transmitted from the parental generation to the progeny or offspring. Similarly, genotype means it is the internal genetic constitution which is responsible for expression of phenotypical character. Like how phenotype means externally visible, genotype means yes, the reason for that phenotype inside the DNA, that is a gene, is known as genotype. This way we are going to use these terminologies oftenly again and again. And a few more terminologies, they are very limited now. And please bear with me. I am going to complete those remaining terminologies. They are like dominant, recessive, dominant gene, recessive gene, then genome, then cross. Here we come across monohybrid, then dihybrid, then P, it is just a letter, we use it for representing certain individuals. Then F, that is F1, F2 and so on. Okay. Let me tell you what do you mean by dominant gene. Here I said already that for the expression of particular trait, we need the chromosomes. I am showing you the chromosomes always in pair here. So as to mention the alleles to express a character. I said, for example, height. To express this particular trait, we require gene on paternal chromosome, father chromosome as well as mother chromosome, paternal chromosome and maternal chromosome. And this is an allele for expressing height, this is an allele for expressing height. If this allele present on paternal chromosome able to suppress a gene present on maternal chromosome to express the character, then it is said to be dominant gene. Let me say height could be expressed either in the form of tallness or darkness. Let us say this is an allele present on paternal chromosome as it is for tallness, and allele present on maternal chromosome is for darkness. Obviously, during reproduction, during fertilization, during the formation of gamete, what happens? They will be recombined, reshuffled, and out of these two, only one will be able to express. The one which is able to express by suppressing the another one for the same character is said to be dominant. And recessive is another kind of gene or allele which gets masked, which gets suppressed by a dominant gene. So because of that reason, the newly born young one would grow taller because gene for tallness is expressed. If that newly born kid will be growing, if it is growing shorter or dwarfer, then it is because of the darkness, allele for darkness. So dominant gene is able to express when it is with another one allele. Then the recessive gene is the one which gets suppressed. But however, recessive gene will also get a chance to express when we will see later on. Then the next one is genome. What do you mean by genome? Genome is the total sum number of chromosomes present in the haploid cell. In a haploid cell or a gamete, that is called as a genome. That means the total number of genes present in male individual are found packaged inside the nucleus of a sperm, and that is called as genetic content of that nucleus in male gamete is known as a genome, that is a human genome. Then the next one is cross. Cross is a deliberate mating, a deliberate sexual intercourse, okay? And this crossing is usually done by doing the experiments on the plants as well as also on the animal species. However, cross or crossing is a kind of deliberative or purposeful mating or purposeful copulation in the living individuals. And based on that, we refer two terminologies. One as monohybrid, one as monohybrid. What is that? Monohybrid cross. Monohybrid cross is a cross, it is a crossing process where single character is taken into consideration. Single character. Each individual may be carrying so many number of characters, but for understanding, for learning purpose, for assessing purpose, if a single character is considered in both male and the female, as I said before, as I referred before, height is a kind of trait. If that single character is referred in crossing experiment, then it is called as a monohybrid cross, whereas a dihybrid means two. Simultaneously, if two characters are considered while crossing the individuals, then it is a dihybrid cross experiment. Monohybrid cross experiment, dihybrid cross experiment, very much popular experiments being carried out by father of genetics, referred as Gregor Johann Mendel. I will talk about Mendel and his contribution to genetics or to the world of genetics in the next segment of this topic. So this is meaning of monohybrid, dihybrid. Next, P for parent. P for parent. We always use a letter, English capital letter P for representing the parent, means both male and the female parents. Then F stands for filial. Filial means the individuals that are filial refers to progeny. Progeny, which is being produced by this parent, That is why I should do like this. Okay. Progeny being produced by this parent is said to be filial generation first time, so it is F1. If the next generation is produced from this F1 generation, then it is said to be second filial generation. Then if we denote as F3, then this F3 is produced by F2. F2 is in turn produced by F1. F1 is being produced by the parent. Like how we represent the crossing experiments by referring this parental generation, F1 generation, F2 generation, and so on. This is how, these are the few terminologies that we come across oftenly while learning each topic, each event that occurs in case of genetics. So that's all. And before concluding this session, I want to mention one more thing, that is homologous chromosomes. You must be knowing it well because you learned while you understood it while learning meiosis kind of cell division. What do you mean by that? Let us say an example of man, human cell. It is let us say it is a diploid cell, maybe hair follicle cell, or a skin cell, or epithelial cell, or a blood cell. You take any kind of diploid cell where it should contain the 2n number of chromosomes, that is equal to 46 chromosomes, right? Please remember, all the 46 chromosomes are not same, out of which 23 are belonging to father and remaining 23 are belonging to mother, paternal, maternal. How to say that out of these 46 chromosomes, these are the homologous chromosomes? Let us consider first chromosome of father and first chromosome of mother, first paternal and maternal chromosome, maybe having its respective size as well as a specific shape. And look at it, I am always showing you the lines, imaginary line, on which I show the presence of alleles of a gene for a particular character. If I say that this first pair of chromosomes are homologous, means they must be similar to each other, but not necessarily be identical, not necessarily be the mirror images, not like that. Totally how many number of genes present on this paternal chromosome on first paternal chromosome must be the same in first maternal chromosome. If I say that here are about 8,000 genes present on first paternal chromosome, which is being paired with the first maternal chromosome, must be also carrying 8,000 genes only. 8,000 genes only. But I am making a point that both this paternal and maternal chromosomes, though these are homologous chromosomes, but they are not mirror images. There are slight differences in the nature of the gene expression, in the nature of the characters they express. That's why, however, these two are referred. Similarly, second pair, third pair, fourth pair, up till 23rd pair. And first 22 pairs of chromosomes, we refer them as autosomes, because they all carry the genes for expressing only the somatic characters. Whereas the last pair, that is 23rd pair, it makes a difference where we call them as allosomes, because they carry less number of genes for somatic characters, but more number of genes for sexual characters. Okay? Sexual characters means sexual dimorphism. This is male and this is female. In this way, we can understand what we mean by homologous chromosomes. And one more terminology, that is homozygous. Homozygous refers to the genes that are similar or identical to each other for expressing the same character. They are said to be homozygous. For example, if I refer if I refer, this is a chromosome, paired chromosomes, paternal, maternal, and here are the alleles of a gene for the same character. To say that these two are homozygous, again considering height character, one type called tall, another type called dwarf. This tall refers to what? The allele of a gene for height present on paternal chromosome, if it is identical to an allele present on the maternal chromosome, then it is said to be homozygous alleles or homozygous to each other. They are identical to each other. For example, tall, if I use a terminology as capital T, usually one more thing I forgot to mention, that for representing the dominant character, dominant allele, we always use English letter that is capital A or B or T or C or D, something like that. While representing the recessive allele, we always use the second order letter, that is a small a, small b, small t, then small c, small d, like this. If we use the second order letter, English letter, then it is for only the recessive. And to represent the dominant allele, we have to use only the English capital letters or first order letters. Okay, and here I am saying that here is an allele for tallness as capital T, and on maternal chromosome also an allele for tallness, that is capital T. Look at it, capital T represents these two are identical to each other. And here we could see the character that is for height as a tallness, but not dwarf. At the same time, it could be expressed in this way, like an allele present on paternal chromosome for tallness is capital T, and another one allele present on maternal chromosome for height as dwarf, small t. Here also we find its phenotype as tall. Capital T represent the homozygous genes and they express the tallness. And one is capital T, another one is small t, as an allele present on paternal chromosome, capital T, and allele present on maternal chromosome as a small t. Still then, since it is a dominant, here we expect the presence, appearance of tallness character. So like how here, if it is similar, it is homozygous. If it is here, it is not similar, this is called heterozygous. Look at it, capital T, small t are not identical. One is dominant and the other is recessive. Paternal gene is dominant and maternal gene is recessive. It is called as heterozygous, but it is tallness because capital T is dominant. Whereas here, capital T, both the alleles for height are for the tallness on maternal chromosome as well as paternal chromosome, then it is said to be homozygous. This way, homozygous, heterozygous condition where the genes are present. And secondly, the homologous chromosomes, I made a mention about it. And lastly, one more terminology, that is locus. Locus is an address of a gene on the chromosome. Locus is a point on chromosome where the gene is located, allele is located, so that position of a gene on the chromosome, position of an allele on the chromosome is known as locus. That's why generally in a layman language we say, what do we mean by locus? Locus is exactly an address of a gene for expressing a specific character in a living individual. This is a way how we can begin to learn the actual fact of genetics hereafter. For your out of interest, I tell you the things like the 2n number of chromosomes in case of man. 2n is equal to 46. Any cell carrying 46 chromosomes must be belonging to man only. Please remember, That means chromosome number is highly specific to each species. Species specific chromosome number we find. In case of man, it is 46. 2n is equal to, I am telling you the paired total number of chromosomes. In case of rana, that is in case of frog, the number of chromosomes is 26. In case of cat, 2n is equal to 38. In case of pig, 2n is equal to 40. In case of monkey, 2n is equal to 48. In case of rat, 2n is equal to 42 chromosomes. In case of horse, 2n is equal to 66 chromosomes. Guinea pig, that is experimental purpose, we use white color or albino rat in the laboratory, as 64 chromosomes. So like how every species animal or every species organism must carry the specific number of chromosomes, whereas the 2n is equal to always in form of pairs, maybe 20 pairs, 10 pairs, 30 pairs, 60 pairs, 40 pairs, something like that. So based on the based on the number of chromosomes, we could be able to assess the living individual cell is belonging to particular species. That's how I think today it is enough to have introduction about the entire field. Today genetics is just not only a subject, it is existing or emerged out as a very new domain where its applications are involved in biotechnology. Look at it, biotechnology is an applied science, whereas genetics is its parent. However, biotechnology students must be very much familiar with all this. Still then for non-biotechnology students, just to refresh the memory, just to have the previous information stored in the mind, or just to recap it, I used all this information, and please consider this information and please be particular and perfect about it. And if you have any doubts or the queries in this regard, in this my first session, please put me questions in the WhatsApp group. As it is mentioned, I am going to upload this entire lecture video into the Google Classroom already. For each class, the registration has been done by our faculties, and just by downloading your Google Classroom, you will get this uploaded lecture video. This is my first session. Please have a look at it. Please listen to my lecture, and if you find difficult in following it, I am ready to change. And if it is very much feasible and convenient for you to understand whatever I spoke about this introduction to genetics topic, that is fine. Okay, and whatever the doubts or the questions, please put me in WhatsApp group for 6th semester students. Okay, each one of you, I hope, will be able to view the uploaded lecture video, and I will come back with the next part of this topic with new information in my next lecture video, that is the second session. Thank you one and all. Take care of yourselves.